Okay, good morning everyone. So today is the 3rd of March 2021 and today's lesson is factors affecting enzyme action. So last lesson we looked at enzymes themselves and did the introduction to them. You'll recognise these learning objectives are the same as last lesson. So you've got to define the term catalyst and enzyme, explain how enzymes work as biological catalysts, and justify the lock and key model relating it to the function of enzymes and explain how temperature and pH can affect the function of enzymes. Your keywords for today are temperature coefficient, denature, optimal temperature, extremophile, polymerase chain reaction, DNA fingerprinting, and genome. I will have these keywords in the assignment brief so that you can look at them there. We've got some of the keywords there, but I'm not going to go through them because we went through these ones last lesson. So I put the ones that you haven't got on this sheet in the assignment brief. Okay, so temperature then. So reactions in cells take place at lower temperatures, for example, 37 degrees Celsius in humans. Enzymes work best at certain temperatures, as we learned last lesson, and that is their optimal temperature. As temperature increases, the general trend is that the rate of reaction also increases. But as enzymes work best at optimal temperatures, if the temperature increases too much, then the enzyme will denature. And that means that it permanently changes shape and it can no longer function anymore because its active site has changed shape and it can't bind to the substrate. And denaturing in human enzymes normally happens at around 40 degrees Celsius. And actually that's true for most organisms. But what about these guys? So these can survive in environments with 1,000 times more radiation, and it says we can even survive in space. So these are little tardigrades, and they are very small little creatures. So they are called tardigrades. They are also called a water bear. They were first identified in 1773. Their size is 0.05 to 1.2 millimeters. So if you get a ruler out, you can look at it and see it is minuscule. They can live pretty much anywhere, but they prefer sediment or wet environments. Their diet, well, that varies per species because there's quite a lot of species of tardigrades. There are, let me just get the figures up. There are over 1,018 species of tardigrades which are currently identified, but there could be more. So depending on the species, some will eat moss, lichen and moss fluid, and some eat other tardigrades, so they are cannibalistic. Reproduction again depends on the species of tardigrade, but they reproduce sexually or asexually. Temperatures they can live in range from between minus 200 degrees Celsius to 148. Apologies for the plane going over. Okay, so temperatures they can survive in range from minus 200 degrees Celsius to 148.9 degrees Celsius. They can also survive without any extra protection, radiation, boiling liquids, 6 times the pressure of the deepest part of the ocean, and also the vacuum of space. So they are quite amazing little creatures. Okay, so we went through the tardigrade just a minute ago, but this is another example here. This is the Antarctic krill, and that is also this type of organism. So these are called extremophiles. So they are organisms with the ability to survive and thrive in extreme environments, which include lots of different environments. For example, hydrothermal vents, which are underwater, freshwater alkaline hot springs, anaerobic geothermal mud and soils, acidic sulfur environments, highly saline lakes, such as the Dead Sea, carbonate springs, alkaline soil. Okay, so denaturing, that is when the amino acid chains begin to unravel. So the bonds between the amino acids and between separate protein molecules will break and, well, will begin to break. So the substrate then no longer fits in the enzyme's active site, so it is no longer complementary to the substrate and can no longer form enzyme substrate complexes, so it can no longer act as a catalyst. And that means that the rate of reaction significantly decreases and even stops if there's no enzyme left. So here is an example of an enzyme and its optimal temperature. So you can see it rises up as the temperature increases, and it keeps rising up until this point here. So this is probably around 35 degrees Celsius, and between 35 and sort of 38, that's the optimal temperature that the reaction works as fast as possible. So that is the enzyme's optimal temperature. And then you get to around 40, 41, and that is when the enzyme starts to denature and the amino acids start to unravel. So you see this sudden drop in rate of reaction, and at about 55 degrees Celsius, the enzyme has denatured and stops working completely. So that is why it's so dangerous for ill people to have high temperatures for an elevated amount of time, for a lengthy amount of time. So we have to reduce the temperature to this optimal range, so that yes, it will be high enough to kill the bacteria, but not high enough to kill the person as well. Okay, so if we look at pH, then similarly to temperature, all enzymes will have an optimal pH, which we'll get to in a minute. So the shape of the active site is due to the bonding and intermolecular forces holding the amino acids together. Changes in pH affect these bonds, which changes the shape of the molecule. So the active site changes shape and it is no longer complementary to the substrate, so it can no longer form enzyme substrate complexes and it doesn't work anymore. This changes the specific shape of the active site and the enzyme is denatured. This is very similar to temperature, as I just said, because they work best at an optimal pH level. Okay, so what we'll do now is look at some questions together. So we've got 3 questions here. First one is 3 marks, second and third are 4 marks. This one you might recognise because I talked about it a little bit earlier about having a high temperature when you're ill. So the first one is describe the effect of temperature on an enzyme-controlled reaction. Use this figure to help you. Explain the effect of temperature on enzyme action. And when you get an infectious disease, you might get a temperature. This is the way your body defends you, as many microorganisms cannot reproduce at higher temperatures. However, people always try to bring the temperature of an ill person down. Explain why this might be the case. Okay, so let's go through the answers then. So, for a describe question, I'm just going to put this out there. You need to say what you see. So describe literally means say what you see. So what is going on in this graph? So as the temperature increases, the rate of reaction also increases. You can see that the rate of reaction at the side here is going up as the temperature increases. But that is up until around 35 degrees Celsius. That is when the enzyme reaches its optimal temperature and the rate of reaction is at its most efficient / or fastest point. So after around 40 degrees Celsius, the rate of reaction decreases as the enzyme's optimal temperature is passed. The rate of reaction rapidly decreases here until around 55 degrees Celsius when the enzyme has completely denatured and no longer works. So that is your describe question, or how you would get the marks for that question there. So let's look at the explain question. So this is your analysis question. So you have to look into why this is happening. So why is the thing that you looked at in the describe question happening here? So as temperature increases, the enzyme and substrate molecules collide more frequently because they have an increased kinetic energy. So as temperature increases, they have more kinetic energy, so they collide more frequently. And that means that there is a higher chance of having more successful collisions taking place. So more enzyme substrate complexes are formed and more products are formed as a result. But again, after around 40 degrees Celsius, the enzyme's bonding starts to be disrupted by the temperature and they begin to break. And this changes the shape of the active site. active site. At around 55 degrees Celsius, the enzyme's active site has completely changed shape permanently, and the active site of the enzyme is no longer complementary to the substrate, and so enzyme-substrate complexes cannot be formed, and this stops the rate of reaction. For the third question, when you get an infectious disease, you might get a temperature. This is the way your body defends you, as many microorganisms cannot reproduce at higher temperatures. However, people always try to bring the temperature of an ill person down. Explain why this might be the case. So you've got an explain question again, so that's another analysis question. So you need to analyse why you think this might be, relating it to what you know about enzymes and their denaturing. So a temperature of more than 40 degrees Celsius is when the active site of this enzyme in particular starts to change shape. So less enzyme-substrate complexes can be formed, and this can hinder the recovery of an ill person, as their body has to work twice as hard to maintain the rate of reaction. Remember, without enzymes, you can't survive. You need to have enzymes to carry out the rate of reaction and increase the rate of reaction for everything, for your metabolism, because without them you will not survive. So lowering the temperature of an ill person to within the safe zone, I've put it in sort of inverted commas here, which is the optimal temperature of the enzyme. Most enzymes in the body will allow the person's immune system to, or of most enzymes in the body, would allow the person's immune system to deal with the microorganism without putting the individual's life at risk due to a high temperature. So lowering it to sort of 38, 39 rather than allowing it to get past this point will still kill the microorganisms because it's at a higher temperature, but it will not put the person's life at risk. So the last thing I want you to do is to look at this. So I'm going to attach some exam questions to the assignment brief. There are 45 questions, so don't do them all. Do questions 1 to 20 of this multiple choice question selection, if you like. But yeah, do exam questions 1 to 20 for this, and then submit your work afterwards. Next lesson is going to be a lesson on graphs, so we're going to swerve a little bit off course of the enzymes, but we are going to relate it later on to pH of enzymes. So I'll see you next lesson.